हेलो गाइस, आई एम शुभम फ्रॉम शुभ ट्यूटोरियल। हम डीबीटी के PYQs सॉल्व कर रहे हैं ऑफ द ईयर 2023 एंड दिस इज द थर्ड वीडियो ऑफ दिस सीरीज। So before we start, subscribe to the channel and press the bell icon to get notified whenever the video is out. This is absolutely free from your side, but this will help us grow. The first question for today is, which one of the following is not true regarding phosphoenol pyruvate carboxykinase type of C4 photosynthesis? Okay, photosynthesis, as I said, बहुत important topic है, right? Now not true यहाँ पे पूछा है. Okay, so what are the statements? Aspartate is transported from mesophyll cells to bundle sheath cells. This is correct. Alanine is transported from bundle sheath cell to mesophyll cell. Oxaloacetate is formed both in mesophyll cell to mesophyll cell and bundle sheath cell. Okay, so this is wrong. mesophyll cell and bundle sheath cell दोनों में ये फॉर्म नहीं होता. Right? Malate is transported from mesophyll cell to bundle sheath cell. Yes. So these are correct statement. Third statement wrong है. अब यहाँ पे picture I have taken. You can save this by removing the screenshot. Okay. तो ऐसे कुछ important जो pictures हैं वो बहुत important होते हैं and this will help you during exam, right? So this is of the C4 pathway. Okay, you can see ये mesophyll cell है and ये bundle sheath cell है, right? Now यहाँ से what we can see, oxaloacetate से malate, malate is coming in bundle sheath cell, right? Malate से pyruvate, pyruvate is going in mesophyll cell. तो बेसिकली यहाँ पे जो क्वेश्चन है इसमें स्टेटमेंट क्या है, oxaloacetate is formed both in mesophyll cell and bundle sheath cell. ऐसा नहीं होता. That we can clearly understand. Oxaloacetate is formed in mesophyll cell only. Right? So this is the oxaloacetate. Oxaloacetate is converted into malate, where in mesophyll cells. Okay, and then this will get transported into bundle sheath cell. So 1, 2, and 4. These are correct statements. And third is the incorrect statement, which is the answer. Because यहाँ पे not true पूछा है. Right? Question you have to understand carefully. Next question. Which one of the following is a neurodegenerative disease? Okay. Now यहाँ पे options है hepatitis, Huntington, osteoporosis, sarcoma. Huntington. Option number 2. Hepatitis is a liver infection. Right? Osteoporosis is a bone disorder. Sarcoma is a cancer of connective tissue. Huntington is a neurodegenerative disease. So easy. डायरेक्ट क्वेश्चंस आते हैं डीबीटी में. इफ यू नो आंसर यू कैन अंडरस्टैंड और यू कैन गिव इट डायरेक्टली, राइट? Cholesterol is transported in blood in the form of low density lipoprotein, right? LDL particles that contain apolipoprotein, cholesterol, cholesterol esters with long fatty acid chain, and phospholipid. Where do you think the phospholipids are located in the LDL particles? So LDL particles में phospholipids का location क्या है? Buried in the core, on the surface, in the lipid bilayer, interacting with hydrophilic region of apolipoprotein. So if you know about LDL structure, you can answer this clearly. That is on the surface. Okay, on the surface, phospholipids are located. See this picture of the lipoprotein. You can see ये जो blue color का हमें दिख रहा है, that is phospholipid on the surface. अंदर कोलेस्टेरॉल ईस्टर है एंड ट्राइग्लिसराइड्स है, राइट? यू कैन सी दिस ग्रीन मॉलिक्यूल इज अ अपोलिपोप्रोटीन. फ्री कोलेस्ट्रॉल भी प्रेजेंट है along with phospholipid, right? Next question. Given below are the two statements. CPG islands are sequences present in eukaryotic DNA. Promoter regions of many expressed genes in eukaryotes are generally enriched in the CPG islands. CPG islands are a sequence present in eukaryotic DNA. This is correct statement. And promoter regions are enriched by the CPG islands of many expressed genes. Yes, both statements correct hai, right? In the light of above statement, choose the correct answer. Both statement 1 and 2 are true. Yes. Statement 1 and statement 2 are false. ऐसा नहीं है. Statement 1 is correct, but statement 2 is false. No. Both the statements are correct. Right? So option number 1 is correct. Which of the following structure is not seen in angiosperms? एंजियोस्पर्म प्लांट्स में से क्या नहीं होता? सेंट्रियोल्स, पेरोक्सिसोम, माइटोकॉन्ड्रिया, गोल्गी कॉम्प्लेक्स. Centrioles. Okay, see, there is one more channel where I cover NCERT syllabus. Okay, NCERT syllabus में अभी सेल का चैप्टर चालू है, तो उसमें ही मैंने अभी कवर किया है. सेंट्रियोल इज द ऑर्गेनल और वी कैन से स्ट्रक्चर जो एनिमल सेल्स में प्रेजेंट होता है. ओके, सेंट्रियोल इज प्रेजेंट ओनली इन एनिमल सेल्स. राइट, उससे ही क्लियरली आंसर आ सकता है. प्लांट्स में सेंट्रियोल्स नहीं होता. पेरोक्सिसोम इज प्रेजेंट, माइटोकॉन्ड्रिया इज प्रेजेंट, गोल्गी कॉम्प्लेक्स इज प्रेजेंट. सेंट्रियोल इज प्रेजेंट ओनली इन एनिमल cells, not in plants. So NCERT also helps, right? CRISPR-Cas9 technology is typically used for genomic sequencing, gene editing, DNA application, gene mapping. So you are updated. अगर आपका molecular biology अच्छा है, and if you know this, this technique is used for gene editing. This is very popular technique. हमने इसका वीडियो भी बनाया है, right? And this is very much used in research nowadays. Okay, this is specifically used for gene editing. Which one of the following is not a type of G protein coupled receptors? Okay, G protein cell signaling, very important, right? Not a type of G protein. GQ, this is a type. GI, this is also type. Okay, inhibitory. GS, stimulatory. We know तीनों भी टाइप्स है. GP, हमने कहीं पे नहीं पढ़ा है. GP. So this is the answer, which is not a type. Okay, so GP is not a type of G protein receptor, right? So fourth option is correct. Again, I have brought this chart. You can make a screenshot. Very important. This is again types of G protein basically. You can see यहाँ पे GS है, GOLF, olfactory, and GI, GO, GT, transducin, right? GQ, G12, 13, the types. One more यहाँ पे important है. See, only selected examples are included. About 20 alpha subunits, at least 6 beta subunits, and 11 gamma subunits have been described in humans. Humans में इतने described है, right? 20 alpha, 6 beta, and 11 gamma subunits. Okay, you can see the functions also. This is a list of some of the important G proteins. Okay, basically these are families, 4 major families of heterotrimeric G protein, right? Which one of the following does not directly account for water potential in plant. See, most of the question यहाँ पे कैसे है, does not, right, जो नहीं करते. तो हमें अगर कंफर्म पता है वाटर पोटेंशियल इज रिलेटेड टू so and so things, ओके, जो हम पढ़ते हैं बुक में, एंड कुछ ऐसे ऑप्शन होते हैं जो हम बुक में पढ़ते ही नहीं, राइट, हमें दिखता ही नहीं है ऐसे कुछ ऑप्शन. So that is confirm our answer. So what water potential is related to pressure, temperature, osmotic compound, gravitation. Water temperature is related to pressure, right? Osmotic compounds also. Okay, we know that water potential जो है that is related to osmotic potential. So yes, this is correct, third option. Gravitation dependent है, temperature हम नहीं पढ़ते, right? So the temperature will be answer. Okay, not we have to find, right? ऐसे हम सॉल्व कर सकते हैं क्वेश्चन. मीन्स हमें देखना है वाटर पोटेंशियल से क्या रिलेटेड है. प्रेशर रिलेटेड है, वी नो. ऑस्मोटिक कंपाउंड रिलेटेड है, ग्रेविटेशन रिलेटेड है, टेम्परेचर इज नॉट रिलेटेड. नेक्स्ट क्वेश्चन, गिवन बिलो आर टू स्टेटमेंट्स, वन इज लेबल्ड एज असर्शन ए, अदर इज लेबल्ड एज रीजन आर. असर्शन मीन्स एक स्टेटमेंट है और उसका रीजन. अ बेस सब्स्टिट्यूशन इन डीएनए डज नॉट ऑलवेज रिजल्ट इन म्यूटेंट फेनोटाइप. ओके, म्यूटेंट फेनोटाइप हमेशा हमें दिखता है, पॉसिबल नहीं है. करेक्ट है. व्हाट इज द रीजन? सम बेस सब्स्टिट्यूशन इन कोडॉन डू नॉट चेंज द एन्कोडेड अमाइनो एसिड. यस, राइट, साइलेंट म्यूटेशन वी कैन से. अगर म्यूटेशन है देन ऑल्सो सेम कोडॉन, सेम अमाइनो एसिड इज एन्कोडेड लाइक दैट. ओके, सो उससे म्यूटेंट फेनोटाइप हमें नहीं दिखता. So both the statements are correct. In the light of above statements, choose the most appropriate answer from the option given below. Both A and R are correct, and R is the correct explanation of A. Yes, first correct है. Both A and R are correct, but R is not the correct. No, correct explanation. A is correct, but R is not correct. A is not correct, but R is correct. So the statements होते हैं from the first option only we can say. And this is about the mutation. Right? One more यहाँ पे पिक्चर मैंने सेलेक्ट किया है, very important. You can make a screenshot. Different types of mutation you can understand with the help of this chart. See, हमारा जो question है, this will be this silent mutation changes a sense codon into a synonymous codon, leaving unchanged the amino acid sequence of the protein. जो अमाइनो एसिड का सीक्वेंस है सेम होता है without any change. So this is a silent mutation. This is the last question for today. Which one of the following laboratory tests would be best to determine the number of CD4 positive cells in the blood of patient infected with HIV? Okay, cell CD4 positive cells we have to determine. Okay. Agglutination, complement fixation, flow cytometry, ELISpot. Cells हमें identify करने हैं. Agglutination will not help. Complementation fixation will not help. Flow cytometry will help. ELISpot will not help again. Right. So specific cells. Okay, marker भी दिया है CD4 plus. This can be determined with flow cytometry technique. Okay, so like this you can solve the question. हमने देखा, if you know, if you have the idea to the concept, you can clearly answer the questions, right? So that's it for this video. I hope this video helps you. If you find this video helpful, do let us know in the comment section. Like this video, share this video with your friends, subscribe to the channel, and stay tuned for the upcoming videos. Thank you.